Okay, for our second didactic of the meeting, we're in for a special treat. Ex-Moffat employee and the only man that's ever attempted an experiment during the IMO workshop, Mehdi Damaghi.
Love you. Thank you so much, Sandy. It's a true pleasure. I mean it. I didn't come back home like after 4 or 5 years, and there was some emotional reason behind it. But I came here for Sandy and Christine, of course, and I'm enjoying it. Today I'm going to talk ecology of breast carcinogenesis, and before that, I just want to tell you where we're coming from. I'm assistant professor, Department of Pathology at the Stony Brook University, and also I'm leading Cancer Evolution Ecology Lab called SEALS. We call ourselves SEALS because SEALs are super cool. And what we are with C. And that's my office, the first arrow. The students get the better of us because they face the amazing view of like the bay touring the Connecticut. But that's fine. So we study evolution, cancer evolution, as we believe that what Toad Abjansi said, nothing in biology makes sense except under the light of evolution. But we also believe what Dr. Grant and Grant said, that nothing in evolutionary biology makes sense except in the light of ecology, evolutionary biology, unless they're under the light of ecology. So to be able to do that, when I started my lab 4 or 5 years ago in Stony Brook, so okay, if we really truly believe in something, we have to work for it, right? So I spent all my startup money, more than $1.5 million, on spatial biology techniques. So you see, we have MALDI, that's a spatial metabolomics, lipidomics, a spatial transcriptomic. I have the cosmics from NanoString in my lab, it's not the core. I have the LCM. You can look at the mutation and genotyping, but spatially. We use the laser, we cut the tissue, and then we sequence it. So anyway, everything about the spatial biology, and the two cancer model we use is breast cancer and ovarian cancer, with two different questions. In breast cancer, the main focus is, okay, why we get cancer at the first point? What happens? Is this a genetic disease? I truly believe not so. I think cancer is not a genetic disease. I said it. I know it's recorded. I wanted to say it and I said it. And I know we can argue about it. In ovarian cancer, which I collaborate with Sandy and Megan, that both are here, we look at mechanism of resistance. Why this nasty disease is so hard to control, and then can we actually find with some evolutionary way to basically come over that resistance? So as Maxi showed, we also do like adaptive therapy, but we also think about like some other evolutionary design treatment that we also use a lot of spatial biology. All right. Today, as I said, I'm going to talk about breast cancer. And I picked one of the selection pressure that is very abundant in many solid tumors, and that's acidosis. So I'll let you guys know where this tumor acidosis come from, why this can apply selection pressure on cancer cells, and how we can actually use that to study the cancer invasion. I love this work. This is started by Martin Corona and the team, and I think that was an eye-opener for many of us. What they did, they used a biopsy needle, they collected tissue from eyelid cancer, right? And they also collected from adjacent normal, and they sequenced it. Guess what they found? They found more mutations sometimes, even several similar mutation in adjacent normal that the cancer cells also have it. So why this adjacent normal are not getting cancer while the other tissue right next to it getting cancer, right? Beautiful. This is also like, came out a couple of years ago. This is in Nature from this Japanese group that they showed several things that I wanted to note basically down. They showed that after menopause and after aging, actually the rate of mutation drops down. This is kind of like, contrary to what we think, that mutations keep accumulating and keep increasing. So the physiology of the human body is important. The other thing they showed, which is crazy interesting, is that every pregnancy drops the mutation 10 times. I still don't understand. I can't wrap my head around it. But that it is there. It's the data there. You can look at the paper. But what I loved about this paper is that at the very end, they said, okay, we don't really understand this, what's going on. because we think microenvironment is important. So I love it. Beautiful. Yes, microenvironment is important, and that's what we have been saying that, for 15 years. I started with Bob Gatenby, Sandy, and Bob Gillies here at Moffitt. And these guys, by the way, I'm like the only one person a part of this. We started digital twin when there is no digital twin. When digital twin. Is that you, Sandy, messaging me? I was trying to give you a compliment that we started digital twin when the digital twin didn't even exist. Right? This is what we started. This is like people like me, experimental biologists, was hired to basically work with mathematicians to basically build this. And now, like NIH, okay, 15 years later, they say, oh, that's important. Sure. Good. So let's talk about breast tumor ecosystem. I move a lot, and this is a little bit. I wish I had the mic that I could move with it. Anyway, uh, as I said, solid tumors are acidic. So as you can see here, this is a breast cancer. And I drew that myself, and I showed schematically how this metabolic phenotype can evolve in breast cancer, right? But this is a ductal carcinoma that can actually be valid for many other tumors, like solid tumors, like prostate, pancreas. Wherever you have duct, this can be actually valid. So as you see all the way in the right, that's a normal physiological homeostatic state. You have epithelial cell, you have a layer of epithelial cell, and then you have that's supported by one layer of my epithelial cells, and you have this blue, basically basement membrane supporting it, and then the ECM and the vasculature. This is all to basically keep the whole system in an equilibrated physiological system, right? What we argue here is that when aging happens, when inflammation happens, then this beautiful equilibrium can be messed up. And that's what happened here. Now you actually see these cells kind of growing inside, growing or kind of like moving inside of whatever because of this ECM is getting aged or the inflammation caused that. So when they go inside, they experience hypoxia. Hypoxia is lack of oxygen, right? And when that happens, the cells have to switch their metabolism from mitochondrial oxidative phosphorylation to glycolysis. Why this is important? It's important because the main source of energy is mitochondria when you have glucose, right? So per 1 mole of glucose, you get 36 mole of ATP. But if there is no oxygen, the cell is still survive. This is an adaptation. That's why you don't see hypoxia as an adaptive selection force. We already adapted to hypoxia. So that's why we kind of like say, okay, you have glucose, you have oxygen, sure, you use mitochondria. If you don't have oxygen, you just switch to glycolysis. The yield of energy is much lower. It's 18 times lower, as you see here. It's like you get 2 mole per 1 mole of glucose. But let's see, let you go. Also, like this slide, I'm kind of like going to gradually introduce you guys to this phenotype. So when you have the oxygen and you use the mitochondria, we call that oxidative phenotype. When you don't have mitochondria and you switch to glycolysis, we call that glycolytic phenotype. And the other phenotype that was discovered by this amazing super genius scientist that called also after him is Warburg phenotype. And this is the guy who wrote this grant, one line, I need 10,000 Swiss or German franc, whatever it was there. And that's how he got it. So what he showed is that he showed that in many proliferative tissue and cancer cells, even in presence of oxygen, that's very important. That's the difference between glycolytic and Warburg phenotype. Even in presence of oxygen, the cancer cells still use glycolysis as one of the main source of energy or some other thing. And this is still like, under debate, like, why the cancer cells would do that, right? But we know that happens. Anyway, so that's it. Getting back to our slide. So as you see here, the cells start getting oxidative. When they get hypoxic, when they experience hypoxia, they switch to glycolysis, right? And the glycolysis, as you see here, the end product of glycolysis is lactic acid. So it is acid. They produce acid, and for the same reason that the oxygen cannot get in, acid cannot go out, right? So these tumors start accumulating acid in that region, in that zone, right? this is a new environment. This is a new physiology. This is a new equilibrium now. Because these cells, what they are used to is like normal pH. Now they're experiencing acid pH, like pH 6.0, pH 6.5, I don't know, whatever it is, but it's new for them. So they have to adapt to it. And that adaptation comes with some new phenotype. That's why I'm start making them different colors and make it colorful. And we think that this adaptation also comes with some other aggressive phenotype, right? That's what some of this is get more invasive, more migratory, and this acid-induced invasion model is introduced. This is actually first, when I came to Moffitt, that was actually introduced to me by Mark Tessie and Sandy through the math model. I was trying to find the math model to put it here, but it was a little bit like old, so I couldn't find it. But that was cool, right? So we wanted to test that. So this is what I started my work at Moffitt 15 years ago. And we start testing all of this selection pressure that cancer cells experience during this process or evolutionary process of from normal to aggressive or metastasis, right? So we put the cells in hypoxia, in acid, in nutrient deprived, in low glucose combination, even starve them to death, and then try to basically mimic what they experience inside the human body, right? So we found several biomarkers, we found some mechanism, which I want to focus on a couple of them. The first one published, almost 10 years ago is that, okay, if the cells are adapting to this acid, they should have different phenotype. Can we test that? Can we actually see if they are different? And if they are, can we actually find the biomarker? Because this is very important to test it later, right? So with a simple experiment, we put the cells, some breast cancer cell line, in normal pH, the other one acid pH, 6.5, and we grow them for like 3 months. And we call them adapted when they basically start growing and then catch the non-adapted cells. So when you put them in acid, they slow down, but they catch up, right? That's important. And then we did the proteomics. This is a mass spec based. We basically look at all the protein using this SciLike, which I'm not going to talk about the details. And then we found some weird result that it took us like almost a year to figure it out. We found many lysosomal protein, particularly LAMP2B, was overexpressed in acid adapted cells. And that was interesting after a while that we basically tried to validate and then find and look at the pathological images. We found that this mechanism that we are showing here, lysosome is the most acidic organelle in our cells, right? The pH can drop to 4.5 to 5, right? And how they protect themselves, they have these highly glycosylated proteins that they kind of make like get hydrated and make like this mucosa, like what you have in your stomach. They kind of like have this little stomach. So they protect themselves, they protect all these enzymes that they don't release them to the cytoplasm and they don't kill themselves. But the other thing that we really liked about this model is that now they turn over, when you put the cells in acid, they use the same mechanism. The extracellular is acidic now, they can use the same protein, make kind of like this, the membrane level like, stronger. But the other thing they do, they release all these hydrolases, and all this make the environment even more acidic. Why is that important? We can talk to Joe Brown about it. This is all about competition, right? You make yourself strong, and then you make your neighbor, the one you're actually competing with, you actually make that one like, easier to kill or easier to die in that very bad environment. So that was nice. We even showed in our tissue that, this can actually be an invasive edge, as you see here. And this was captured beautifully with the model that Jeffrey West, Sandy, and Mark Tessie built it. And I'm trying to catch here is like basically I put these markers, the acid adaptation marker, next to the CA9 marker, which people usually use it as a hypoxia marker, but it's actually acid producing marker. How is acid producing? Because it basically neutralizes the intracellular pH, which is super critical for the cell survival, but they throw out the acid. So it's not a pump, they use the bicarbonate, and they basically acidify their environment. So this is actually producing acid. Think about the same strategy I was telling you. So they actually make more acidic microenvironments, right? So this is cool. All right, the math model also, like, what Jeff and Mark Tessie, actually the head behind this guy's genius. He actually showed that, this phenotype, kind of Warburg phenotype, that cells produce more acid, and at the same time they're also acid resistant. They can be actually at a very invasive edge. And these are the one who basically the pink here, They are actually the one who can actually eventually kill the patient. I said, okay, cool. This is fine. This is all like in 2D Flask. We build this all beautiful model. Is it actually real? Can we actually test this? Can we actually use this in clinic? As a translational scientist, can we actually translate this to our clinic? Cool. So for that reason, we said, okay, if that's the case, let's go look at the very earliest stage of breast cancer, in this case, DCIS, ductal carcinoma in situ, and then see if we can actually find this biomarker related to invasion. How is that important? 85% of women with DCIS, they never met upstage to invasive disease. What does that mean? That means that they have a stable disease that we should not even call it cancer. They stay pure DCIS for the rest of their life. But because of the 15%, the rest of that 15% that the upstage to invasive disease, we treat all of them the same. So we over-treating our patient, right? So that's $1 million question. Can we, but how do you know? How do you know if a patient at the biopsy has like Heiko Steenberg describing that for the radiation project? Can we actually use a biopsy to actually predict? Because that's the most important part, right? It's okay if our hypothesis is true, if this acid-induced invasion model is valid, can we actually use it in this model? So we built this cohort at Moffitt. Excuse me. So these are all the patients that diagnosed with DCIS at the biopsy time. So we only have one biopsy. But the good thing about this model is that we have follow-up surgery. We have follow-up excision. So we know what happened to them, or if a pathology missed, if our biopsy missed the actual invasive node, right? So we collect that biopsy and say, okay, can we actually use this to test our hypothesis? Okay, we did that. We did the sequential cut. We stand for LAMP2, we stand for CA9, the two marker of acid adapted phenotype I tell you. And then we also use H&E image. And the idea is that we really think this is not a marker of like frequency of one cell with some markers that basically should be counted in, like the traditional way that pathologists look at. You should actually look at the patterns. You should look at if in that environment, if in that niche, the cells are actually evolving to an invasive phenotype. So that's why basically we use HE to find this habitat and then find and correlate the cells with CA9 or LAMP2 positive on top of them, and we build these niches, right? So that's a super simple idea, but very laborious, very tedious work that postdocs and to a student actually work on this, right? So the way we did is, okay, first we look at the pathology specimens, we look at the whole study tissue, we do exactly what the pathologists do. We did that, absolutely no difference. None of these markers has any predictive power. But now let's look at the ducts, because each single duct in cancer, now we know that in breast cancer, there's a multicolonic disease, multicolonal disease. They might actually come from different sources. They might actually evolve in different environment. So let's just annotate every single duct. And not just the duct, we also created this habitat zone based on the oxygen diffusion distance that, if it's 100, this is first, I think, published by Rakesh Jain at the Harvard, is 160, 140 mathematical model that we have shows between 125 to 140. So we took the minimum, we built these layers, as you see here. We built these layers. So that's actually, the red is your normoxic zone, that your cells are have different metabolism. You go inside, they experience hypoxia. Of course, there is the markers for necrotic, right? We also look at the reactive extrema because I have an active project with Kajia here, but that's for future. So we did that by just doing it, by just separating tissues to habitat, the simplest model of oxygen habitat. We found some differences now. From absolute zero, we got to some 90%, 94%. Nice. Okay, super cool. Let's look at the niches that we were talking about. So we make the neighborhood based on the CNN expression and LAMP2 expression, and then we basically take those neighborhood, superimpose it on the HE images, and then we extract the features of the HE. And these features can be actually combined with the CNN expression level, and then we call that a niche. And there's billions of features that you can actually come up with, and these are the formula that we use for those niches. We look at the distance of the cells, we look at the empty distance, and also the density and all of that. This is published, you guys can actually look at it. And then we look at the patterns. So if you find these features, fine, you can find any niches, whatever, or patterning amongst all this, tissue, but are they reproducible? So we call patterns, and we escort them. So patterns inside a slide per patient and patterns among all the patients. And with that, we could actually show that if you look at the niches, you can increase the prediction power to 74%. Okay, that's not a crazy number, but that's a number that Jeffrey West, not this Jeffrey, there is a really much older pathologist in Stanford with Michelangelo. They got the same power, prediction power with 50 markers. They have this multiplex IHC that they use this 50 markers. So this is good. We just use 2 markers, the same power. So we are hopeful. Okay, can we do better? Sure. We have the machine in the lab. Let's do the same thing, but with our opinion, with the way that we're doing it. So we have this 50 markers. Now we're looking at all the stroma, all the immune, all the cancer cells, all different type of cells. And we do this neighborhood in a more complex way. We do it for both cancer cells and immune cells and stroma cells, of course. And now about the habitat. For the habitat, now we just, okay, step back. All we did is like the cell phenotyping. Can we also define some metabolic, use some metabolism or some lipidomic to define this habitat? So we do MALDI. So we have the HE and we do the sequential cut. We do MALDI. What is MALDI? As I say, it's a spatial mass spec. How does it work? You shine laser to your tissue. You know the coordinate that you're shining your laser, and it's evaporate the specimen, and then you look at the mass charge ratio. And that's great because you can see in what spot you have what type of metabolite or lipid or protein, whatever you have, right? Super cool. This is just, show that, how we did it. You see like those little dots that you burn the sample. It's not the sample because you apply matrix on top of it. That's why it works. And this is some example from the same tissue I was showing you. And you see we can measure lactate, glucose, which is important for us. G6P is a glucose type that go inside the cell, so it's different. It's more important than free glucose. Excuse me. I wish I had some water, Sandy. Sorry, it's just too much smoke in the air. Yeah, so this is how it goes. You just do the same thing, the clustering that you do with the cells. You do it similar. It's a Leiden clustering. I'm pretty sure all of you guys know much better. Thank you, sir. And it's much better. And we do it. And you see, look at the patterns, right? You can literally see these guys. And this is unbiased. My student gonna be here tomorrow. Nahil is not here, and UJ is doing some internship somewhere in Canada, I guess, but he's gonna be back. They literally do it unbiased. We really don't, I don't give them, and this is good. Working with mathematicians is sometimes really good. They don't, no offense, but they don't know much about the biology most of the time, right? So this is good. It's like, okay, can you just do this? And say, okay, let's see what happens, right? And then they come with this, oh, this is a beautiful pattern. It's like, I love it. That's good. And we do the same thing, as I said, with the multiplex IF. This neighborhood I told you, just gonna go through it really quick. And then we found some cool stuff. We could look at acid lactic niches. It's not published, that's why I'm like going through it really quick. It's just stupid because I know this is recorded. So anyway, so you have these niches of like, you have this metabolic, habitat of like lactic acid reach versus the glucose reach. And then you basically put the other phenotype on top of it, and now you can look at it. This is a collaboration, as I said, with Carja. We're trying to see how this acidosis can actually turn the phenotype of cancer-associated fibroblasts. How they basically, can we actually turn normal fibroblasts to calves, but just changing them from one niche to the other, or habitat to the other habitat? And the other one with Ravi and Vey at the UPenn that we looking at the macrophages. We found some cool phenotype of macrophages related to both DCIS in progression and also the response to PD-L1 therapy. All right. Do I have 5 more minutes? Perfect. Thank you. So this is good because this is really my favorite part, right? This is what me and Sandy kind of like did the very cool collaboration. Of course, this is also like the model is created by Mark Tessie, and I created the spheroid. So you see this half is an actual experiment. The other half is the Sandy's experiment and Mark Test's experiment. So and they look really cool, right? So and the patterns, right? So as I said, like when I moved when I started my own lab, I was like, okay, I wanna spend all my money on ecology. If I play poker, I always go all in, and this is all in. Ecology, tumor ecosystem is the most important thing. We have to, if you believe in it, you go for it, right? So, okay, fine. I've done 2D, it's cool. Question that people also ask Maxi, it's very cool, but it's very hard to basically reproduce in the patient, right? So let's just start with the model. And the first, anytime that you see Sandy start talking, is like, we love models, all the models are good, but let's start like with the simplest model that is not too simple, right? So I think 3D spheroid are those models for us, for biologists. Why? Because you start with the cell, exactly the same cells, and you just let them grow like a tumor, right? And they create their own habitat and their own niche because they just growing, they're getting bigger and bigger, they produce acid, they create hypoxia. You don't have to put them in any of this environment anymore. And we have enough knowledge to know where this guy's coming from, right? So we did that. So, okay, let's grow this spheroid, let's grow thousands of them. And then we basically do what I just showed you that we did with the patient with DCIS, right? We do sequential cut, and we do this multiplex phenotyping. This is again like testing our own hypothesis and our model, like, can we actually find these patterns that the environment basically pose on the cells, right? So we did that, and when you basically run MALDI on them, look at these beautiful images. Just look at the patterns, right? You don't really need to know what molecules these guys are coming from. Some of them are like more at the edge, some of them are centered, some of them in between, right? This is amazing, right? Of course, we can actually look at the biology and see like, does that actually make sense? Yeah, look at the lactate. It's in the hypoxic acidic zone. Look at the glucose, G6P, is in the normoxic zone. How does that make sense? Because the cells in the center, they're turning into lactate in a much faster rate. The cells in normoxic zone, no. They don't need it because it's also coming from the media around them, right? Super cool. But we run two mode of lipidomics, a small molecule. That's my student who just made it, Sandy. I wasn't lying. You're stuck in JFK airport for 24 hours. And then we made this beautiful the 3D layers of metabolomics, lipidomics, two modes, right? And again, unbiased, unsupervised clustering. Look at the patterns again, right? You see this beautiful orange at the edge, and then you have blue, and then you have green in the center, which is cool. And then it's okay, how do we know if this is actually related to the phenotype that we are interested? The cool thing is that we have this panel of all this metabolic phenotype that have been working past 15 years in a multiplex panel now. We have these 40 markers that you can look at the glucose deprivation. If these guys need to use glucose, they use GLUT1, CA9, and all of that, right? So you see, there is a model. This is a that we can actually call the phenotype of these cells. Now we have the metabolite and lipid of those at the same spheroid. You put them on top of each other, and you can actually test if some specific phenotype coming from the same some location. And what we found is our acid zone that we think they actually correlate with a lot of sphingolipid. I even didn't, to be honest, like 4 years ago when we started, I didn't know even the sphingolipid, has some biological role. But when you go read about them, you actually see, yes, they are. So go back, okay, let's just do a sanity check. Do they actually go up when you put the cells in acid? Yes, we did a regular mass spec. We put our cells in acid, the sphingolipid, ceramide goes up, they shoots up. And then, of course, we wanted to check that if that's really happening and if this cancer cells fitness under acid. So we did a CRISPR-Cas. I just summarize this for you guys. You don't have to look at all those pathways. There's like 2 production paths for ceramide and the 4 exit paths for it. And then we did CRISPR-Cas, and we found that there's several genes in all those 4 paths of ceramide clearance involved in acid response. I was like, okay, fine, this is cool. I can validate all of this, but do they actually matter if you actually treat your spheroid when you inhibit this tissue? Because the spheroid is a 3D model we want to check. And when we did, they didn't really do it. They actually helped the spheroid grow better. And these are the drugs that they are trying to basically help our patient to treat patient. You give them a S1P inhibitor, they actually grow better, right? So it's okay, this is crazy. What if we caught all 4 heads of like this ceramide production? Do we actually kill the cells? Yes, we do. This is all, you see, when you grow the steroid, when you only cut all those different paths of like production ceramide, they die. So we think this is plasticity. We double check it with our molding model, and you actually see here, it just showed, trust me, I can describe it later, that when you cut one head, they actually put all the ceramide into the other path. So see, sometimes you can actually like help the cancer cells to find a better direction. All right, that was cool. And then we also measure fitness, which is important. And with that, I would love to thank. And that's it. Thank you, Sandy. Thank you, my team. These are the guys who did the work and the funding.
Okay, thanks, Mehdi. That was quite the tour de force. There was a lot to digest there. Does anybody have a quick question for Mehdi?
Yeah, great. Great and comprehensive talk, Mehdi. And I have a question about that last part, the lipids and the overall lipid metabolism. So you begin with lysosomes and actually their distribution or increase in acidosis. So, I mean, maybe there is more in the story, but it is known that those lipid species are important for membrane generation, right? And those membranes are important for the organelle biology as well.
Yeah.
So maybe one by one, they don't have too much role, but did you check if organelle biology distribution frequency, do they change?
Very good question. Actually, the lysosome, I have a PhD. I'm chairing the committee of PhD student from Yusuf Hannoun, who's actually who's the one who actually discovered the sphingolipid. He's actually looking at that after this finding, and he found super cool result. He's gonna publish soon. But as you said, also like a sphingolipid have very major role in membrane stability, and that's actually when I show the last slide about the fitness, that gives the highest fitness. That means that being essential sometimes is more important than just giving you some advantage. Okay, we can talk later after. Thank you, Sandy.
If you've got more questions for Mehdi, we can ask at the break. So thanks a lot. Okay.